Good evening and welcome to everybody for this webinar being organized by NephroPlus on this exciting new topic of xenotransplantation. Our host for the evening and the moderator for the discussion is Dr. Vivekanand Jha, who's the Executive Director of the George Institute of Public Health and also the Chairman of NephroPlus's Scientific Advisory Board. I'll hand it over to you, Dr. Jha, to take the proceedings forward. Thank you so much.
Thank you very much, Kamal. Thank you everyone for joining and welcome to another of these regular webinars, which we do from time to time with the idea of identifying and discussing some interesting, not only interesting, but also topics that are potentially very, very important for our patients, if not for now, for future. Today, we are going to discuss one such topic, which is xenotransplantation. You all must have been aware of the news that we hear about xenotransplantation and the increasing successes of xenotransplantation, which are extremely, extremely exciting. So, to discuss xenotransplantation, we have none other than Dr. Vasishta Tatapudi, from New York University Langone hospitals. He is affiliated with, of course, the Langone hospitals. He did his MBBS from Jipmer, Pondicherry, and since then he went on to receive his training in the US and has been in this program for a long time now. So he has been associated with the xenotransplantation program at the hospital for a long time, and now really is well known for his contributions to the program. He has written papers in multiple high-impact journals, and it is now my pleasure to not stand between him and all of you anymore and invite him to talk about xenotransplantation. Where are we? Is it close to coming to the clinic, or are we still too far? So over to you, Dr. Tatapudi.
Thank you, Dr. Jha, and all the other panelists as well. Thank you for the invitation and that very generous introduction as well. I think I'll start by stating the objectives of my talk. My goal is to review the rationale for xenotransplantation. Why is it necessary? I'll review some of the immunology behind xenotransplantation, the genetic modifications and the immunosuppressive regimens that would underpin xenotransplantation when it comes to the clinic. And I'll talk about the trajectory of the improvement in renal xenograft survival in preclinical studies that have led up to some of the human studies that we're now doing. I'll briefly discuss give you an overview of the recent and rapid developments in pick-to-human xenotransplantation, and I'll allude to some of the clinical trials that are on the horizon. And then I guess we can have a discussion afterwards. I'd like to start with stating the magnitude of the problem that xenotransplantation aims to address. So this is data from the US, and I'm sure it reflects data in India and across the world. This is really a global problem, the problem of organ shortage. It is a public health crisis. And in the US, there are about 80,000 patients on the kidney transplant waiting list. And unfortunately, we're only able to do about 25,000 kidney transplants. Again, this is in the US, 25,000 per year. And after being on the wait list for 5 years, only about half the patients receive a kidney transplant. About 1/3 either die or they're removed from the waiting list because their health has deteriorated. And another 20% are still waiting for a kidney transplant. And as a result, in the US, about 7,000 patients actually die every year waiting for a kidney transplant. And so what is xenotransplant? Xenotransplantation is the transplantation of organs, tissues, or even cells from a non-human animal source into a human. And this is potentially a solution to the pressing problem of organ shortage. And theoretically, it can provide an unlimited source of organs. And that's why some view it as a sort of a silver bullet. But I'll hope to convince you that we're closer, that's closer to reality by the end of the talk. And here's a timeline of events, if you will. Attempts to perform xenotransplants have been around for nearly a century now. Pig or goat kidneys were attached to the blood vessels of human recipients in the arm, but these survived only for a few days. This was back in the early 1900s. And these attempts were not successful because of hyperacute rejection due to cross-species immunologic incompatibility. And primates, baboons, monkeys, they're evolutionarily, they're closer to humans. And so in the 1960s, here in New York, there was a surgeon who performed a xenotransplant from a chimpanzee to a human, a female patient suffering from kidney failure. She received a chimpanzee kidney and survived for 9 months thereafter, and then succumbed to an electrolyte abnormality. It was an acute event that led to her death. But since then, the field of xenotransplant has moved away from using primate-sourced organs, and mainly for ethical reasons, but also for some technical reasons that I'll talk about. And so today, pigs are considered a suitable organ source, and there are a number of reasons. Primary among them is that the organ size and physiology, they match that of human. They breed rapidly and attain large litter sizes. So if you want to scale xenotransplantation to match the need, you have to have supply of organs. And so this is important. Pigs can be genetically engineered with relative ease compared to a lot of other species, at least using current technologies. And in the US and in several countries in the world, pigs are consumed as a source of food and also, more importantly, medicinal products. So at least in the current ethical framework in the US and maybe in a lot of other countries, it is considered acceptable to use pigs. But why do we need to genetically engineer these pig donors and not simply use wild-type pig organs? Well, that's because what I'm showing you here is what would occur if you took a wild-type or unmodified pig kidney and implanted it into a non-human primate, like a baboon or a chimpanzee or a monkey. What you're seeing here is on the left, a kidney that's being implanted, and on the right is that same kidney a few minutes after anastomosis. And this is hyperacute rejection. You see that kidney there is mottled and necrotic and needs to come out. This is similar to what would happen in a hyperacute rejection in allotransplantation, where preformed anti-HLA antibodies, if they were present in the recipient serum, would bind to HLA antigens in the transplanted kidney, activate complement, and a host of other cascades, including the thrombotic cascade, coagulation cascade, and cause graft destruction. And so in an allotransplant, and I don't need to repeat this to this audience, we avoid this hyperacute rejection by performing a prospective cross-match to screen for pre-existing anti-HLA antibodies. But pigs don't express HLA because they're not human. They don't express human leukocyte antigens. They express swine leukocyte antigens. The question then is, why are these wild-type pig xenografts rejected immediately if pigs don't even express HLA? And so, that's because the cell surface proteins on pigs and many organisms, they're heavily glycosylated. And if you'll indulge me, I'll go into, some of the mechanistic aspects of what leads to hyperacute rejection in pig to primate transplants. So pigs, like organisms across the animal kingdom, they have heavily glycosylated cell surface proteins. The sugar component of these glycoproteins, they're polysaccharide molecules. They're called glycans. And these glycans are highly polymorphic, and their structure depends on glycosylase enzymes. And these glycans, they coat the cell surface like a dense vegetation. They're everywhere. And so this brings me to what's known as the alpha-gal epitope. Alpha-gal is one such glycan. It exists in most mammals. It's formed by the addition of a galactose molecule in an alpha-1,3 linkage to other galactose molecules that make up glycans. And so, and the enzymes responsible for the alpha-gal epitope are known as alpha-1,3 galactosyltransferases. So alpha-gal coats the cell surfaces of many, many organisms. Bacteria express alpha-gal, so do pigs. But during evolution of primates and humans about 30 million years ago, this enzyme, alpha-1,3 galactosyltransferase, was inactivated due to a frameshift mutation. So it turns out that chimpanzees, baboons, monkeys, and humans don't express alpha-gal. So we lack this epitope. And we've known this since the 1980s. But humans, we're exposed to alpha-gal, even though we don't express it on our cell surface, we're exposed to it right from infancy due to the presence of alpha-gal epitopes on bacterial cell surfaces in the gut microbiome. So we develop naturally occurring anti-alpha-gal antibodies. So alpha-gal becomes the primary xenoantigen that elicits hyperacute rejection. And that's where genetic engineering has been revolutionary. What we can now do is generate alpha-gal knockout pigs that lack alpha-gal on their pig kidneys. And so they look less foreign to the human immune system. And so this is what would happen if you transplanted a pig kidney expressing alpha-gal into a human or a primate. This is hyperacute rejection. I showed you a similar cartoon earlier, but this here shows alpha-gal antibodies directed towards the alpha-gal epitope that's present on the microvasculature in the pig kidney, again, would elicit responses that are akin to hyperacute rejection and cause graft destruction. So when you use alpha-gal knockout pig kidneys, in theory, these can be produced using a combination of somatic cell nuclear transfer technology that was used, that's been used for cloning several animals, most famously Dolly the sheep back in the 1990s. So this is cloning technology. You combine that with CRISPR-Cas9 gene editing technology that was developed in the last decade or so. And so you combine those two technologies to now genetically edit pigs to knock out alpha-gal. So even though we've known about the importance of this epitope since the late 1980s, it's only in the last several years that we've been able to clone and breed alpha-gal knockout pigs at scale to be able to do these studies. And in addition to alpha-gal, there are two other carbohydrate xenoantigens that have been identified that are also important. These are New5GC and SDA, and the relevant enzymes are SEMA and beta-4-galin-T2. And pigs that, in which one, two, or all three of these xenoantigens have been knocked out have been generated by a number of biotech companies now. And in addition to knocking out those three glycans that I've just mentioned, we can also delete or insert other genes that may be clinically relevant, like complement regulatory genes and human transgenes, so they can be inserted into the pigs to regulate complement and coagulation cascade that are important in the immune responses that might occur after a xenotransplant. And so with the advent of all of these sophisticated genetic engineering techniques, and as well as better immunosuppression in the last 2 or 3 decades, survival of genetically modified pig xenografts in non-human primate models. So these are pig to primate or baboon or monkey xenotransplants that have been done by various groups in the US and in Europe for the last 3 years, attempting to make xenotransplantation a clinical reality for humans. And all of these studies, you see steady improvement in pig xenograft survival, almost an exponential increase in the last 15 years or so with the advent of CRISPR gene editing technology. And I'll show you a couple of these non-human primate studies. This is a study from Dr. Sykes and David Sachs's group in New York. This was a study back in the early 2000s. This was a big step forward when graft survival went from a few days to more than 3 months of survival in a non-human primate model. This is another study by Dr. David Cooper's group showing xenograft survival for up to 136 days in baboons using immunosuppression known as anti-CD40 antibody. This is some histology there. So you can see at day 103, histology looks relatively normal when the graft was still functioning. And by day 136, when the graft was lost, there was a rejection. And this is another study by Dr. Adams and Tector, who are now able to get survival up to 400 days using double knockout pigs and anti-CD154 based immunosuppression. So this is 2018. And all of these studies led the FDA in the US to approve alpha-gal knockout pigs for consumption as food and also for potential therapeutic uses. And these alpha-gal knockout pigs are produced now by several biotech companies in the US. But going from these preclinical studies to clinical studies in humans is a giant leap. So going from pig to baboon studies to pig to human studies is a giant leap. So that's where the decedent xenotransplant model comes in. It's an intermediate step. Here we transplant pig kidneys into decedents who are recently brain-dead individuals maintained on ventilator support in the ICU. So this allows us to test genetic engineering and immunosuppression protocols without risks to live humans. And so this is what we did in 2021 at NYU, led by Dr. Montgomery, who's the director of our Transplant Institute here, and really the visionary behind this. Who kickstarted pig-to-human xenotransplantation, moved the emphasis from non-human primate studies that have been done for the last 2 or 3 decades to really focus the attention on moving it to the clinic. And so in 2021, we identified a brain-dead individual who was not a candidate for organ donation, so they couldn't donate their kidneys and liver and lungs. And consent was obtained from the decedent's next of kin. The decedent was transferred to a purpose-built ICU. And decedent is a term, again, I want to emphasize, is something we're using to describe a brain-dead individual who would receive a pig kidney transplant. The pig kidney was procured from a pathogen-free facility, and these were alpha-gal knockout pigs. These were thymokidneys. This is a thymokidney is when a pig thymus is implanted under the renal capsule of the pig, and I'll talk about this later if we have time. And this is done to induce potentially tolerance towards the pig donor. And our surgeons procured the kidneys from a donor pig in a pathogen-free facility, airlifted the organ to New York City, and performed the transplant. So this is not unlike how a deceased donor transplant would occur. You procure the organ, transport it, and then implant it. The renal artery in this case was, and the vein were anastomosed to the femoral blood vessels. The kidney was, it remained outside the body on the thigh for direct observation for signs of hyperacute rejection, and also to facilitate serial biopsies. The ureter was cannulated and connected to an external urinary drainage collection system. The native kidneys in this case were not removed. The kidney was perfused only for 54 hours before we concluded the study. And this is because that's the permission we received from regulatory agencies at that point. And we conducted 2 such xenotransplants in September and November 2021. And the purpose of these studies were to demonstrate that knocking out alpha-gal would prevent a hyperacute rejection in a pig-to-human xenotransplant. So our goal was limited. We only wanted to show that by knocking out alpha-gal, we could prevent a hyperacute rejection. These are some of the results. The right panel, there shows data from the first. The left panel shows data from the first xenograft, and the right one from the second xenograft. The x-axis is time from transplant. The dotted vertical line there is the time of transplant, and the blue and the red bars are native and xenograft urine output. And the black graph there shows you the kinetic estimated GFR. So you can see that immediately after xenotransplant, the graft began to produce urine. Those are the red bars, significant urine output, exceeding sometimes 500 ml per hour. And the kinetic estimated GFR actually increased after xenotransplant. So even though there was some native kidney function, the GFR increased further after xenotransplant. And these are biopsies that we performed after the xenotransplant at various time points. And the histology that I'm showing you here was at the time that the study was concluded at 54 hours. These are normal appearing glomeruli with open capillary loops showing no microvascular inflammation, and the tubular interstitium there showing no evidence of lymphoplasmacytic infiltration. So no hyperacute rejection. This is immunofluorescence, the first xenograft showing negative complement C4d. There was some mild complement activation in the second xenograft, but it didn't result in other histologic features of rejection, and certainly there was no impact on xenograft function as we measured the urine output. This is electron microscopy. I'm pointing to podocyte foot processes, which look healthy. There's no foot process effacement. We didn't observe any proteinuria in the short duration of this study. And so a similar study was conducted by Dr. Jamie Locke's group at the University of Alabama soon after our first decedent xenotransplant. And there were some key differences between their study and ours at NYU. They used pig kidneys with 10 gene edits. Both native kidneys were removed in the study, and they implanted 2 pig kidneys in the abdomen. And the good news is that they also didn't observe hyperacute rejection. In their study, though, the creatinine continued to rise after xenotransplant for the duration of the study. They studied the xenografts for 72 hours. They observed some thrombotic microangiopathy on the biopsy and complement deposition. So they concluded that the thrombotic microangiopathy that they observed was due to complement activation by innate immune mechanisms. So they learned from that, and they conducted another study where they implanted a 10 gene edited pig kidney into a decedent, this time observed it for 7 days, and they had added a complement blocker, eculizumab this time. And with that, they were able to eliminate thrombotic microangiopathy, and they observed excellent xenograft function. And I'm showing you the trend in creatinine there and the creatinine clearance. And by day 7, you can see there that the creatinine was 0.9. And following these 2 studies at NYU and at Alabama, a group in Maryland, again here in the US, led by Dr. Mohammed Mohyuddin and Dr. Bart Griffith, who's the surgeon, performed the first ever pig to living human heart transplant using a 10 gene edited heart. And the recipient in this case survived for 2 months I'm showing you the picture there in the corner of Dr. Griffith, the surgeon, and the recipient who survived for 2 months with a functioning cardiac xenotransplant. And then after that, at NYU, we conducted 2 more pig to decedent cardiac xenotransplants into decedents. So these are, again, brain-dead individuals receiving cardiac xenotransplants. And we demonstrated the absence of hyperacute rejection. What you're seeing here is pig hearts beating in human chests of decedents. And I don't know if there's any cardiologists in the audience, but these are echocardiograms of these pig hearts showing good function. So this was in 2022. But after these studies, what we were left with is little knowledge about what the adaptive immune responses are that would develop in the days and weeks and months after xenotransplant, because our studies, both at NYU and Alabama, were short duration, 54 hours, 72 hours, 1 week. So in July 2023, we performed another decedent xenotransplant study at NYU. This time, the decedent received an alpha-gal knockout pig kidney, and we observed the decedent for 61 days in an ICU on a ventilator. And this decedent received induction and maintenance immunosuppression consisting of thymoglobulin, tacrolimus, mycophenolate, steroids, and in addition, we used rituximab, which is a B-cell depleting agent, belatacept, a co-stimulation blocker, and eculizumab, a complement C5 inhibitor. And what I'm showing you here is a prospective cross-match that we performed looking for IgG and IgM antibodies against donor lymphocytes. And we showed with serial dilution that this cross-match was negative, and then we proceeded to transplant. Again, this is a post-reperfusion biopsy that we performed, showing no evidence of microvascular inflammation. And I have to say, this, to me, looks almost indistinguishable from a human kidney. Just by looking at histology here, you can't tell whether this is a pig kidney or a human kidney. Again, no microvascular inflammation, no glomerulitis or peritubular capillaritis, no interstitial inflammation. So therefore, again, no hyperacute rejection. We observed excellent xenograft function throughout the first month, and at day 28, the serum creatinine was in fact 0.4, as you can see in the graph here, shown there in blue. We also measured cystatin C, BUN. We performed 24-hour urine creatinine clearance measurements. All of these were consistent with excellent xenograft function. And as I said, the creatinine was 0.4. We performed several protocol biopsies during the first month. Histology looked normal, no evidence of microvascular inflammation. But on immunofluorescence, we were beginning to see some complement deposition. What I'm showing you there is C1q, C4d, C3, all part of the complement cascade. So these were demonstrable on these biopsies. But we weren't seeing any histologic evidence of microvascular inflammation, and no clinical evidence of rejection either. We suspect because our complement blocker, eculizumab, was effective in blocking terminal complement. We also saw some IgM deposition, not so much IgG. And then around day 33, we began to see subtle changes, a slight increase in creatinine, still within the normal range, but the creatinine went up to 0.7. There was a slight decrease in urine output. And one of the advantages of the decedent model is that it allows you to sample tissue and blood at frequent intervals. And so we were able to perform biopsies whenever there were subtle changes. And we did one more again, day 33. And this time, we found mild glomerulitis, peritubular capillaritis, and some mesangiolisis indicative of endothelial injury. And the immunofluorescence actually showed at this stage C4d positivity in the peritubular capillaries, not just in the glomerulus. So these were all features that are suggestive of antibody-mediated rejection. And so at this stage, we also had access to cultured donor pig aortic endothelial cells because we had a month. We were able to obtain these cells in culture. And then so we went back and performed cross-match, flow cytometry cross-match using these aortic endothelial cells. And we found that these cross-matches were indicative of some reactivity against donor porcine aortic endothelial cells, even though the cross-match initially was negative towards lymphocytes. And so we thought all of these fit with antibody-mediated rejection, and we treated this episode of rejection with plasma exchange and also the addition of a complement C3 blocker this time, not a C5 agent, which we were using. So now we decided to block the complement cascade a little more upstream at the level of C3 using this drug called pegcetacoplan. And many of you may be familiar. This is a drug that's now being tested for other glomerular diseases. It's actually FDA approved in the US for paroxysmal nocturnal hemoglobinuria. So this is a drug with a safety, a track record of safety. And so we used it to block C3. And then we began to see an immediate response. The creatinine improved back to baseline. The urine output increased. And then we continued the study for another nearly 30 days. And you're seeing their trends in eGFR, creatinine clearance, serum creatinine, all normalized. And by the time we concluded the study at day 61, the serum creatinine was 0.4. And again, we performed a biopsy at explant. You're seeing a glomerulus there with resolution of the microvascular inflammation and tubular interstitium looking normal, healthy tubular epithelial cells there on the right. So what we learned from this long-term decedent study now was that, again, alpha-gal knockout is sufficient to prevent hyperacute rejection. Naturally occurring IgG or IgM antibodies could cause antibody-mediated rejection, and we were able to successfully treat that with plasmapheresis and an addition of a complement blocker. And all of these studies, both primate and human decedent studies, led the FDA to grant us permission on a compassionate use basis to perform the first xenotransplant in living human recipients. And you may have heard about these events in the news. The first ever case was performed on March 16, 2024 at the Massachusetts General Hospital in Boston. And the second case was performed at NYU in April 2024. These are the pioneering patients who received those xenotransplants. The MGH patient, their experience was actually published in the New England Journal recently. And their recipient was a 62-year-old with a prior failed transplant, had a history of coronary disease. He received a 69 gene-edited pig kidney. And all of these, gene edits are, I think we don't have time to go over all of them. But what they also did was knocked out certain endogenous retroviruses to prevent viral transmission. Here's the immunosuppressive protocol that they used. This is the graft function that they observed. This patient was on dialysis, came off dialysis immediately. The creatinine improved, as you can see there. This patient had an episode of T-cell mediated rejection on postoperative day 8, which they were successfully able to treat and reverse, and ended up having good graft function with a creatinine of about 1.5 to 2 for nearly 2 months. But unfortunately, this patient died due to a sudden cardiac event on postoperative day 52, but with a functioning xenograft at day 52. This is some histology there. Day 8 biopsy showing immunohistochemistry there in the middle panel, middle column there. You can see infiltrating immune cells. They were able to treat that, goes away, and then relatively normal looking histology on day 52. This is the xenotransplant in the living recipient that we performed at NYU. We presented this data recently at a national transplant conference. This patient was a complicated individual. She had heart failure, she had kidney failure, was on dialysis, but she was too sick to receive a heart and a kidney transplant from an allo transplant. And so we received permission from the FDA to perform an LVAD procedure first, so a left ventricular assist device, following which we performed implanted a pig kidney in her. And so it's a complicated patient. She did do well early on. Extensive immunosuppression here using standard immunosuppressive agents in addition to C3 and C5 blockers. And as I'm showing you here, excellent xenograft function initially for the first month. Serum creatinine there in green was less than 1 up until 1 month. And then she developed certain infectious complications. As I said, she had an LVAD. She also developed bacteremia, line sepsis perhaps, became hemodynamically unstable, and then developed graft dysfunction. But we believe she didn't have rejection. What you're seeing there at the end around postoperative day 42 is graft injury due to acute tubular necrosis and infarction rather than rejection. Which brings me closer to the present day. Looking at all of this data, the non-human primate data, the decedent studies that I went over, and also the other 2 living recipients, the FDA gave permission to 2 more patients to receive pig kidneys. And so we performed another pig to living human recipient kidney transplant at NYU in late 2024. That's the patient in the middle there, and flanked by our surgeons, Dr. Montgomery and Dr. Locke, who performed the transplant, looking well, the patient looking well there after xenotransplant. And this is the other patient who received a xenotransplant at the Mass General Hospital in January 2025. Both of these patients had excellent xenotransplant function immediately after xenotransplant. This is limited data that I'm showing you from our patient at NYU because we haven't published this data yet. She was on dialysis. She was, in fact, a living donor back in the 1990s. She donated a kidney to her mother, then developed preeclampsia during a pregnancy afterwards, and progressed to end-stage kidney disease. She was highly sensitized to HLA, and so she was waiting for a kidney transplant, desperate, for 8 years, and then we performed a xenotransplant. Immediately came off dialysis. The creatinine normalized. She had excellent xenotransplant for 4 months, and then unfortunately had a rejection. And this is in the news. We had to explant that kidney. She's now back on dialysis, but now we've been able to achieve 4 months of graft function in a living recipient. And at least up until that point, this was the longest known xenograft survival in a human recipient. I will tell you that the recipient at Mass General Hospital, as of a couple of weeks ago, is still doing well. I don't have all of the information, but that patient is still alive with a functioning xenograft. And with all of this information, the FDA has again reviewed all of this information and has felt confident enough that we are now at a stage where we can proceed to clinical trials in humans. They have greenlit 2 clinical trials. One of these trials will be performed at NYU and Johns Hopkins, and another trial will be performed at the Mass General Hospital. And so these trials are on the horizon. We're in the process of determining who the candidates are for these studies. And so these will not be one-off cases. These will be multiple patients enrolled in clinical trials, and will give us an opportunity to assess the safety and the efficacy of xenotransplantation in living humans now at scale. And so I think there are still some knowledge gaps that remain. And as nephrologists, we're all interested in knowing whether these xenografts will, provide function. What about electrolyte and acid-base homeostasis? There are certain important and interesting physiologic mismatches that could arise after xenotransplant. For example, it's known that pig renin doesn't break down human angiotensinogen. So are there going to be problems associated with the integrity of the renin-angiotensin-aldosterone axis after xenotransplant? Another interesting thing is that pig ADH, antidiuretic hormone, is lysine vasopressin as opposed to human ADH, which is arginine vasopressin. So the receptors are slightly different. Are we going to see nephrogenic diabetes insipidus after a xenotransplant? These are questions that need to be answered. What are the optimal gene edits? Is it 1 gene edit? Is it 10 gene edits? Is it 69 gene edits? Not clear yet. What are the molecular mechanisms of xenograft injury that would occur after a xenotransplant? What are the optimal cross-match techniques? Should we use lymphocytes for cross-match? Should we use aortic endothelial cells? And a really important question to address is transmission of zoonotic infections. I will tell you that so far in all the xenotransplants in humans that have been done so far, there has been no evidence of zoonotic infection transmission, but surveillance is going to be really important, and also selecting pig donors that are negative for pre-existing infections to the extent possible is important. Cross-sensitization, is that going to occur? Cross-sensitization essentially alludes to, if you do a xenotransplant, is the recipient going to develop anti-HLA antibodies that is going to make it difficult for them to receive an allotransplant should the xenotransplant fail? I believe that this is not the case. Cross-sensitization doesn't seem to occur, but there's some conflicting data regarding this. So I think clinical trials are going to be important, and support for this from the National Institutes of Health through funding is going to be important as well. So this is what's on the horizon. This is what we need to understand before I think we can say that this is safe and efficacious and can be scaled to patients across the world. I would want to acknowledge the team here at NYU. Again, I want to shout out to Dr. Robert Montgomery, the visionary behind all of this at NYU, who's there in the center there with the rest of our team members. And I'm happy to take any questions.
Thank you. Thank you very much, Dr. Tatapudi, for giving us the tour de force and taking us on a journey of xenotransplantation over the course of the years. And this is the most recent journey that we have had. As a concept, xenotransplantation has been around for a long time, but it took all this time and perhaps for you to come into the team for us to get a xenotransplantation as to the clinic as we have it today. So thank you very much. And we have now, if you could convert the view to a panel view, whoever is managing the technical. Yes, great, thank you. We now have a number of expert members of the panel with us who will carry the discussion forward, because this brings to light a number of important clinical issues that are relevant and come to the mind of nephrologists. These are the kind of questions that our patients obviously ask us, and wherever they see the news that comes now in news channels, but increasingly on Google searches, hope dawns, and what are the various things that they hear? What are the issues from their perspective, and what do the practicing nephrologists, transplant surgeons, and also the patients be mindful of? How do we pass on this message to our patients in a way that is consistent and it sort of gives them a realistic current information? and where do we expect this revolutionary technique to be? So can I introduce Professor Vijay Kher, who is currently he's held multiple positions and is currently at the Epitome Hospitals in New Delhi. He has been a leader in Indian nephrology and transplantation world for 50 years or more. So welcome, Professor Kher. We have
Thank
Professor
you.
N. Gopalakrishnan from till recently of Madras Medical College, and he has also been an institution builder at Madras Medical College. He established the Institute for Kidney Disease and Transplantation. This is an institute of excellence and research there. And recently, he is superannuated, but he remains active in clinical medicine, teaching, and mentoring students. Then we have Dr. Sukanya Govindan. Dr. Sukanya Govindan is a pediatric nephrologist. This technique is of special relevance to the pediatric population because transplantation is something that that obviously is the most suitable option of kidney replacement therapy to all children with end-stage kidney failure. And they have a very long life to look forward to. So how can xenotransplantation really help them? And last but not least, we have Dr. Khalid Al-Hassan from Saudi Arabia. Dr. Khalid Al-Hassan is a leader of the Saudi Society of Nephrology, and he's not a stranger to Indian nephrology community. And Saudi Arabia also has been a leader in providing kidney replacement therapy services to its citizens and has taken a leadership role in a number of initiatives, which include deceased donor organ transplantation, dialysis, and we'll really want to hear from Dr. Khalid, his view also in how this technique will probably apply there. So as Dr. Tatapudi, you highlighted, and obviously after going through all the developments, you highlighted in your last couple of slides there, outstanding questions also, and the challenges that we discussed. And so starting from how the use of dissident model helped in generating very, very useful knowledge to the concept of thymokidney that you brought in, and perhaps we could discuss that a little more as to what does that mean. Gene knockouts, how many genes need to be knocked out? That's an important question. I don't know. You said that this is still open in terms of the number of genes that need to be knocked out. The zoonosis risk thresholds, what are those? And as you said, that the MGH program knocked out a number of viruses, genes that manage susceptibility to porcine viruses. So these are a few questions, but then there are many other questions also which we shall discuss. So I'll now open this conversation to the panel and invite all the panelists to make comments, starting from you, Professor Kher.
Yeah, I think I must first of all thank you for including me in the panel. I don't have any knowledge about this except that what you read in the newspaper, journals, and what Dr. Tatapudi spoke. Excellent overview of the current standing of xenotransplantation as it stands, and also the very good prospective news, which I didn't know that much about, is the 2 trials which the FDA has approved to be done. These may be small trials, they may not be yet large trials. I learned that probably one trial has to include some smaller number of patients, the other is somewhere around 50 transplants. Maybe he can clarify it later as to what it is. But I think there is huge interest amongst the population. As a clinician, when I sit in the OPD, every time there is a news about xenotransplantation in the newspaper, my patients come with the clips or send me WhatsApp clips about when is it going to come and what it looks. Even people who are still patients with chronic kidney disease are not yet either requiring dialysis, but they know that they will develop end-stage kidney disease at some point of time, and probably may need transplant. So they're very keen to find out as to when is it going to be available and all that. So at the moment, obviously, you can't say that it is yet there for the clinic, for the patient as yet. The other question which I will have for Dr. Tatapudi will be also the possible costs, because these costs are going to be enormous to start with. So whether will any democratization ever happen if it comes to the clinic as such, and that would be an important question as well. The other thing I have is one other question is, does the modified big kidney produce erythropoietin and increases the hemoglobin in these patients, or is this erythropoietin somewhat different? And what is the hemoglobin level in the patients which have survived, say, for 2 months, 6 months, or 4 months?
Thank you, Doctor. I think we can we can invite first comments from all the panelists and maybe take up some of all the questions together, if that's all right with you, Dr. Tatapudi. So next, I'll pass on to you, Professor Gopalakrishnan, from your perspective of what Dr. Tatapudi has shared with us.
First of all, I'm like everyone, I'm truly excited. What looked almost impossible till a decade ago, now it is becoming a kind of reality, even though it is difficult to say that it has already reached the clinic, but definitely the progress so far that has been achieved itself is quite exciting. My question will be, what do you think is the minimum number of gene edits? On the one end, just single gene knockout. On the other end, so far 69 gene edits. So what will be the kind of via media? And do you feel that despite the 3 carbohydrate gene knockouts, antibody-mediated rejection is happening and endothelial injury is happening, probably do you think it is mandatory that in addition to that, the gene edits have to be done with regard to genes which are the regulating complement activation? So that is first question. The second question is that you have put it in the last slide. I'll elaborate on that. So I learned that the lifespan of pigs is around 10-15 years. So after the program becomes a successful one, do you think the biological senescence of the graft that is bound to happen, and so will it have a finite life? It may not be compatible with the human life expectancy. And the third thing is that, as Professor Jas asked for, is thymokidney is a kind of a game changer? Let me limit with these questions, and if time permits, the second round, I'll ask other questions.
Thank you, Dr. Balakrishnan. As I said, this kind of technique is especially important for children. And so I'd like to hear from you, Dr. Govindan, your perspectives, and what are the questions that come to your mind?
Yeah, first of all, thanks for including me in this panel, and it's really exciting to look forward to xenotransplants as one of the options for our patients. And especially with regards to pediatric patients, as also Dr. Gopalakrishnan said, the longevity of the graft is a question. And also pig kidneys can have lower nephron mass compared to, human kidneys. And that would affect the longevity, or would the age of the porcine kidney, at what time they are being implanted? Do they have any adaptation after, being implanted into a human being? So we do look at, fetal kidneys or younger donors' kidneys being implanted into children and adults. And so that would be a consideration in pediatrics. And also, are there any surgical differences in these patients with multiple renal arteries or stuff? Because already pediatric transplants can be surgically challenging for many reasons. And would these also add on? And also, I was wondering whether, because of the difference in the xenoantigens or the repertoire of antigens that these porcine kidneys would introduce us to, are these rejection episodes only limited to renal effects, or do we also see systemic effects when a rejection episode happens? So those would be some of the discussion points that I would like to be addressed. Thank you.
Thank you, Sukanya. From your perspective, Dr. Khalid Al Hassan, how does this how is this technology perceived in the part of the world where you live and work? Obviously, we hear about it in conferences, read about it in journals like New England Journal of Medicine. Every time there is a case, it is sure to get into New England Journal. So how do you perceive it?
Yeah, absolutely. Initially, I would like to thank you, Vivek, for inviting me to be panelist, and really, I highly interested and enjoyed the talk by our speaker Tatapudi for really very terrific talk and excellent overview. Actually, I remember we invited Professor Montgomery, and he came here in Riyadh on March 24. And he gave one talk about the xenotransplantation, which was at that time, 1.5 years back. It was just in the beginning, and especially started the clinical trials. So obviously, not only the transplant physician, as also the patient, they are desperately looking for an extra thing comes to the donor bank. We are a huge shortage in all over the world for the organs. So I totally agree with the first couple of slides by our speakers when he talked about the huge shortage. That's an excellent door that can be open to find organ for our community. I just told my colleague before we start opening this webinar that we have done a survey to the healthcare workers, just not yet published, about the xenotransplantation and what they think about it. In addition, of course, to different population, to other population, the ethical and religion part is play, I think, an important thing. Why choosing pig, not other than pig, other than orc, or what other animals than it? I think there is a couple of idea came from the survey that people are thinking about stigmatism of the patient, to stigmata he is having an animal organs and going with animal organ. Any psychological part can affected him and discussed feelings and the other thing. But definitely it is one of the doors that can open. My questions to our speaker is actually he very nicely talk about the gaps that need to be known. What do you think from all those gaps that really will be one of the big challenging to us in future?
Great, thank you, Khalid. And before I pass on to you, Dr. Tatapudi, there is also a question in the question and answer panel from Dr. Sonal Taguditi, and she asked a series of raises a series of issues, some which have been already pointed out, but I think other issues also. What are the regulatory challenges, and in addition to the kind of issues that Dr. Khalid has raised about the use of certain species for certain populations in terms of acceptance, both social acceptance and but religious issues. Are there other ethical challenges as well? So I'll just pass on to you first to maybe respond to some of these issues and expand on anything else that you might have thought.
Thank you. I think that was, I think between all of you, you've brought up some excellent and important questions. I'll see if I can address each of them. Maybe I'll start with what you alluded to first, Dr. Jha. I think the thymokidney, I only briefly talked about it during the talk, but if I may, I can share another image. Let me know if you see this. Do you see this?
Yeah.
Yes.
Okay, yeah, so this is just from a publication that just came out from our group. I thought it'd be useful to explain what the thymokidney is. Essentially, we take the pig's thymus and implant it under the thymus is ground up, minced, and then a portion of it is implanted under the renal capsule months before the organ is actually procured for donation. And the rationale is that this pig thymus, which reconstitutes under the renal capsule, will educate the recipient's immune system by only selecting lymphocytes that are not reactive to the donor organ, and prevent donor reactive cells from causing a rejection. So that's the idea behind the thymokidney. Obviously, a lot still needs to be learned about this, but this is essentially a tool for tolerance. The studies that we did, the short-term decedent studies back in 2021, didn't allow us to study any of these adaptive immune responses because they were really short in duration. But the longer-term decedent study, where we kept the decedent maintain the decedent on life support for 61 days, did allow us to study whether the thymokidney was in fact effective in producing tolerance. We believe it does. We haven't published that data yet, but I do believe that there will be a positive impact with using a thymokidney rather than just a gene-edited pig kidney. So that's, I think, addressing your question. I think Dr. Kerr had brought up clinical trials, and we're all excited that these are now on the horizon. You are correct, even though you mentioned that you're only hearing about this in the news. I think you know a lot about this. You're correct. There are two trials. The one at Mass General Hospital will enroll 6 patients, a smaller trial. They're using 69 gene-edited pig kidneys. And the trial that we will be part of here at NYU, which will initially start off with enrollment of 6 patients between NYU and Hopkins, Johns Hopkins. And then the FDA will review those results, and if they're acceptable, we will proceed to a multi-center enrollment of up to 50 patients. So this would be a larger trial. There are slight differences in the genetic edits that would be used in our trial. We would use 10 gene-edited pig kidneys. Most of those 69 gene edits are in fact CRISPR knockouts of porcine endogenous retroviruses. We share the 10 gene edits that the pigs that would be used at our trial are almost identical to the gene edits that would be used in the MGH trial. There's only one transgene that's different, but most of the differences come from knockouts of porcine endogenous retroviruses. The importance of doing that, I think, is yet to be seen, because we haven't seen any transmission of retroviruses, porcine endogenous retroviruses, to date. We think that the risk of transmission is really low. So then the question arises, do we really need to do we need to include so many gene edits? The more gene edits you introduce, the chances of off-target effects are greater too, but it's yet to be known. Your question about EPO, I think, is well taken. What we know so far is that there is some degree of homology between pig and human EPO. So it's possible that the EPO that is produced by the pig kidney will lead to hematopoiesis. But in the studies that we've done so far, again, unpublished, we are yet to determine whether that is the case. So I don't have an answer to that question yet. But I will say, though, that with the availability of recombinant EPO, it may not be such a big deal. I don't think it's one of the physiologic mismatches that will lead to a huge challenge. Cost is an important question. Again, at this stage, there is no price tag on these kidneys. I'm sure in the early stages, the price tag will be high. It's not going to be a situation where patients can pay out of pocket, and this is going to be different in different countries and how this will be reimbursed. But the goal, I believe, from all of these biotech companies that are making these organs, these pigs, is to scale it to the extent that costs will come down. Pigs are initially cloned, and cloning is expensive, but then they can be bred. Once they start breeding and attain large litter sizes, costs should come down. So that's my answer about cost. I think if I want to go down the list, I think Dr. Gopalakrishnan had asked about the minimum number of gene edits. I think we were able to demonstrate that just knocking out alpha-gal will prevent hyperacute rejection. We observed an episode of rejection about 1 month after the decedent xenotransplant that we were successfully able to treat and reverse. So it's possible that just one is enough. A parsimonious model would suffice. But I think a case can be made for more edits. So I think we need to figure that out both in the clinical trials, but also in more decedent studies that we're hoping to do at NYU. More long-term decedent studies, the ones that I described earlier. We're hoping to undertake more of those if we have enough resources to do that. And I think the decedent model will allow us to test various permutations and combinations of gene edits and figure out how few or how many we need. Other epitopes that might elicit rejection. Are there other genes that we need to delete? I think we need to figure out why are we seeing the rejection episode that we did see? What are the molecular mechanisms? I think some studies are needed to figure that out. And I think if we can figure out the molecular mechanism, then you can design the appropriate targeted therapies as well. Lifespan and senescence, I think it's, again, an excellent question. Doesn't always come up, but I think you brought it up. I think it's relevant, but as things stand right now, allograft survival in the US is about 15 to 20 years. So I think if we can get even a decade out of a pig kidney, that would still, in my mind, be an acceptable result. I think Dr. Govindan had asked about, how the implications for pediatric transplantation. You were also interested in longevity. I hope I answered that question to a certain extent. Nephron mass, it's unclear, exactly what the glomerular density is in these pig kidneys. It could also vary from the type of pig kidney we use. Maybe it's different if you use an alpha-gal knockout versus a 10 gene edit versus a 69 gene edit. So we're trying to study that. What is the nephron mass of each of these kidneys? But in terms of GFR, we've seen that we've used 10 gene edits and we've used alpha-gal knockout alone. We've seen excellent GFR from just one kidney. So I don't think it will be a huge problem in terms of providing adequate GFR. These kidneys do grow after implantation, and that's why the 10 gene edits, one of the gene edits is a growth hormone receptor knockout. So those kidneys don't grow as much. I think that will be relevant to pediatric transplantation because in an adult abdomen, even if the kidney grows, you're not worried about compartment syndrome and things like that. Growth of the organ does become relevant to pediatric heart transplantation because it's a confined cavity. So that's certainly important. I think there are some surgical differences. You were interested in the anatomy. And also, I believe the anatomy might differ in based on the number of gene edits. That's just anecdotal information that our surgeons have relayed to me. They think some of the vascular anatomy looks different depending on the number of gene edits, but not to the extent that it increases risk of surgical complications. Systemic effects of rejection, I don't think we observed anything. Our living patient did have a rejection Eventually, she required a xenograft nephrectomy, but she's doing well. Certainly, I mean, infection with heavy immunosuppression is possible, so there could be that systemic effect. I think Dr. Al-Hassan and I were talking about this earlier before we started the talk. I'm eager to see what the results of your survey are, the details of that. There are certainly ethical and religious considerations. I think those are going to be really important. They will differ from place to place, country to country. And I think surveys like yours are going to be really important in determining acceptability when you go from place to place. Psychological issues, how patients perceive this. My experience has been that, these patients who are on dialysis, especially, and their families, and they are eager to get this off the ground. So they're not, I think my perception is that they're not averse to receiving pig organs. But again, individuals differ, cultures differ. That's really important. This cannot be imposed on people who don't want it. Yeah, I think there was a question in the chat about other regulatory challenges, what the FDA is looking for in the US, what regulatory agencies in other countries will look for. At least in the US, I can tell you they were confident enough that the results so far warranted clinical trials. So I think that's a positive sign. What they look for in the trials is standard stuff, safety and efficacy. Efficacy will have to do with providing adequate GFR, ensuring that there are no physiologic incompatibilities like electrolyte, acid base, and abnormalities related to mismatch between pig and human renin, ADH, things like that, and certainly zoonotic infection transmission. That's going to be really important to surveil. Are there other ethical challenges? I'm sure there could be, but the perception in the U.S., I can tell you, has been overwhelmingly positive so far. That doesn't mean that there aren't skeptics, but I think that's why the clinical trials will be important, and we'll have to see how things advance in the next months and years to come.
Great. Thank you. I think I'm pretty sure that all of these questions have covered a range of issues and you have answered them beautifully, Dr. Tatapudi, and then every answer has enhanced the knowledge of the audience here. I think if I may just make two final points and invite your response to that. One is that you described the use of the cultured aortic endothelial cells for cross-patch, and that introduces perhaps some novel immunological mechanisms and possible way of monitoring the immunological response in these individuals, and that might lead to some modification in range of tests that might need to be done. And the second one I might ask is if you can share some light on, you did talk about the principles of these clinical trials that will be conducted now with safety and efficacy. But are there any specific trial design related issues that they may still be in development? So to the extent that you can share those with us, I think it'll be great to know. Because other than the primary endpoints, which are likely to be much more obvious, safety and efficacy, there could be a number of secondary endpoints that you have in mind, or exploratory endpoints, which will really advance the frontiers of knowledge. So to the extent that you can share anything about those with us, that would be great. Thank you.
Yeah, no, again, thank you for the insightful questions. I think the aortic endothelial cells, using those versus, PBMCs or lymphocytes is an important question that we need to figure out. We certainly saw a difference in cross-match strength when we used endothelial cells versus lymphocytes. And it's possible that some epitopes that are expressed on endothelial cells are not expressed on lymphocytes, and that's why the difference occurs in the cross-match strength. And it's possible that the aortic endothelial cells are better surrogates for what would occur after xenotransplant. But the challenge is, as I mentioned, that you don't have access to these cells at the time of transplant. You can't prospectively have access to these cultured cells. But it's also possible that you can use surrogate cells from other pigs with a similar genetic background. So then you can have those cells available to you pre-transplant, and they might indicate what would happen if you proceeded with the transplant. So that's something we're looking into. The other thing is it better to use, renal, cortical endothelial cells? Those are also cells that we could, obtain. And so exactly what type of cell should we use for the cross-match is yet to be determined. It's simple enough in an allotransplant, but maybe there's more nuance to this in a xenotransplant setting. So I think that's a great question. I don't have all the answers, but we're, I think we and other groups are looking into it. Trial design, what I can tell you and what is publicly known is that the trial that we would be part of here at NYU and at Hopkins is a quote unquote a phaseless study. Essentially, it's not phase 3, 2, or 1. It's an adaptive type of design which will allow us to change our protocol as the study progresses. And as I said, we will initially enroll 6 patients. The data will be reviewed, and then it'll be a multi-center trial. There are certain specific inclusion and exclusion criteria where there's an age range for the recruited patients between 55 and 70. There are certain criteria which the FDA is interested in. They don't, we don't want to be enrolling patients whose expected survival is really poor, but at the same time, we don't want to be enrolling patients who would do well with an allotransplant. So we want to be targeted. There are certain parameters that we would use for this, like the estimated post-transplant survival, the EPTS score, which is a risk, a tool that allows us to calculate post-transplant survival. It needs to be within a certain range for the patients to qualify for the trial, things like that, that we're going to use. And I think some of this information will become publicly available in the next few months as the trial begins to enroll.
Wonderful. Thank you very much. We have, I think, one more question. Dr. Moderator asks, can the gene editing technology be transferred to India for local development? I think the answer should be yes to this. Technology transfer is not impossible, but employing it at scale is going to be the challenge. But Dr. Tatapudi, your thoughts?
Yeah, I think this is the question that I encounter most often. Scalability, and I think it's probably the most important question to address globally if this needs to be a success. I agree with you. I think it should be possible to transfer the technology. Perhaps it could be done in a staged manner. Maybe initially the goal would be to perhaps transport the kidney on a perfusion device and do the transplant in India or other countries. And then if that's successful, think about, are there resources and that either the government or private industry would avail for us to be able to build out an enterprise and pathogen-free facilities and things like that in each country to make it available to patients who need it.
Yeah. All right, thank you. I think we have gone over time already. Part of it was my fault because I came in late. But I think part of it was because really of the engrossing nature of the topic and the way you covered it. You covered almost everything and shared with us a lot of new knowledge. So thank you once again, Dr. Tatapudi. And thank you all also to all our panelists for stimulating the discussion and bringing out important topics that help us think creatively about these new technologies. And whenever a new technology comes, there is always an issue of how will this new technology be implemented in any resource poor setting like in India, and that's something we'll have to continue to watch for. But it is our job as advocates for our patients to work with all stakeholders, including governments, including private industry, and including with colleagues such as you. I think you are our ambassadors as very important intellectual contributor to the diaspora to bring this new knowledge back to the country. So I hope that we are in exciting times. So we'll see. Is dialysis going to go away in the next 10 or 20 years? I don't know. I don't think so. But still, these are exciting times and full of hope for our patients. So thank you very much. And I'd like, before closing and passing on to Kamal, I just want to express all my thanks to the panelists.
Thank you so much, Dr. Jha. Thank you so much for moderating and hosting this webinar. Thank you, Dr. Tatapudi, for your excellent talk. Thank you, Dr. Kher, Dr. Govindan, Dr. Gopal Krishnan, and Dr. Al Hassan for participating in today's talk. On behalf of the entire NephroPlus team, I'd also like to thank all the attendees for joining today's webinar. Thank you. Good evening.
All right. Thank you.
Thank
Bye.
you. Good evening.